Well, good morning, ladies and gentlemen, and welcome to the first BOFIN webinar of 2023. My name's Tom Allen-Stevens. I lead the British On-Farm Innovation Network, and I cannot tell you how excited I am about what we have in store for you today. But before I introduce today's main event, I'd just like to give you a bit of a preview of some of the other exciting projects we have coming up this year. So I'm just going to share my screen. I hope you can all see that. So suffice it to say, we've been pretty busy of late looking for fascinating projects that will make a positive difference to how we farm and how we can co-create and shape new developments and technologies as they come along. Now, the good news is that there's no shortage of people who want to work with you, which is no surprise because you are the best farmers in the world. So I hope many of you are now familiar with the slug-resistant wheat project. So this is a project that got underway last June and revolves around field trials of Watkins 788 that was identified. This is a landrace variety that was identified in feeding trials at John Innes Centre as one that was consistently spurned by slugs. Now, only field trials will confirm if there is a slug-resistant trait that could help farmers move away from a reliance on chemical control measures and the potential environmental impact of those chemicals. So you stepped up to the mark, and we now have 10 slug sleuths conducting those very trials on the first ever plots of Watkins 788 to be grown on British farms. And I'm delighted to say that the Environment Agency has very kindly come forward and supported the trial. So that's allowed us to conduct more feeding trials at John Innes Centre, which are underway at the moment. And you may have seen that we are appealing for slug scouts to trap and send in slugs for the trials. So please do get involved. And there's more information on the website about this. So the sleuths are also coming together for a workshop. And then this will be followed on the 16th of March with a webinar to reveal the initial results of the trial. So please put this date in your diary. The other good news is that we've secured funding for a second year of trials, both feeding trials and field trials, thanks to a grant from ycorrhizal. And then we've been talking with RAGT about insecticide-free farming. Now, this is only really possible thanks to new genetics coming forward, and particularly BYDV resistance. And they're keen to work with you and to understand how we can really make the most of these genetics. And of course, the new £45 a hectare payment under SFI for not applying pesticides, insecticides, I mean. So the provisional date for a webinar on this is the 20th of April. Look out for more on this and how you can be involved. And in fact, we plan to have a webinar on the third Thursday of every month this year. So why not book that in your diary now? And then there's harvest weed seed control. And thank you to those of you who have got involved and followed the fortunes of the 3 weed seed pioneers who have Redicop seed mills fitted to their combines. And we've been working with NIAB on assessing the amount of grassweed seeds still standing at harvest and available to the unit, as well as the efficacy of the SCU itself. And I'm delighted to say that we've secured funding again through UKRI for a second year of trials. And this is where we'll be looking for seed scouts to get involved in some groundbreaking research. So more on this later, and look out for a webinar, which provisional date for that is planned for May. Now, last but by no means least, BOFIN is part of a large industry consortium led by PGRO, and we have a very ambitious and exciting project due to get underway shortly. Now, look out for more on this one when details are announced, and we're planning a kickoff webinar in June. We're looking for more opportunities as well. So I'll bring we're looking for more opportunities as well for fascinating projects to get involved in. So I'll bring more on this at the end of this morning's webinar. But look, let's move on to today's main event. So here's the agenda. I'm going to shut up in a minute and hand over to Dr. Tom Thurkel, who's going to bring you some insight into his research. And I have to say, it's fascinating stuff. It really is. And he's also going to be telling us about this project that I told you about just recently and give us some more details. We're then delighted to say we're going to be joined by Simon Cowell and Tim Parton, who many of you will know are part of the Green Farm Collective. They've been doing some quite a lot of work over the years off their own bat on soil mycorrhizal fungi. So we'll hear a bit more about that from them after that. And then we're going to open it up into a general discussion. Now, you'll see at the bottom of the screen, there should be a Q&A and a chat button. So do please go in there and let's get the chat going. Let's get some questions going as well. We will look at all of those comments and chat and every question as well. If we don't answer the questions, then I will feed them back and we'll try and get some answers to you. What I'm hoping to do during the discussion this time, which is be different from previous BOFIN webinars, is to actually bring you into the discussion itself. So if you've got a specific question when we get into the discussion, then if you stick your hand up, and I'll try and actually bring you in so that you can turn your video on and so on, as long as you want to, of course. Whether that'll work, I don't know. Zoom is quite a funny thing when it comes to these sorts of things. So anyway, listen, without further ado, I'm going to hand over to Tom. So Tom, would you like to turn on your video and join us? Turn on your video and unmute yourself. Now, Tom, I don't know if you've seen, but there is an article in CPM magazine in the February issue on this as well. Yeah, written by quite a talented journalist, I understand. And I have to say, it was one of the most fascinating articles I've ever written. So listen, I'm going to hand over to Tom now. So do you want to take things away, Tom?
Yep, will do. Thank you very much.
I'll just stop sharing my screen.
Okay, hopefully everybody can see this. So thank you everybody for coming. It's great to see so many of you here. And I'm going to be talking about my research and, as Tom says, a project I'd like to get off the ground looking at fungal communities in crops. And what we're talking about here is the arbuscular mycorrhizal fungi. And it's this association between plant roots and soil fungi. And the fungi grow into the roots and form these exchange structures you can hopefully see here that have been, we've dug up plant roots and selectively stained the fungal tissue here. So all of this in blue is fungal tissue inside the roots. And these structures allow the fungus to exchange nutrients with the plants. And in addition to growing inside the roots, they're also growing out through the soil. And you can see here the external hyphae of this fungi growing out from the root. And the symbiosis is really important for plant nutrient uptake, and it can also be beneficial to the plant in other ways. So plants that have got a lot of mycorrhizas can have improved defense responses to pests and pathogens, their water use efficiency can be better, and they can also have tolerance to heavy metals. And sort of looking more broadly, soils that are rich in these fungi often have better soil structure and can even have reduced greenhouse gas emissions. And it's often been said that plants don't have roots, they have mycorrhizas. And I think it's fair to say this because of how abundant and sort of widespread these fungi are. Pretty much all of our crop plants are mycorrhizal, all of the cereals. It's really just the brassicas that don't form these associations. And mycorrhizal fungi are basically everywhere in arable soils, but we're not sure what they're doing. We know that you plant your cereals, whatever it is, wheat, barley, or your beans, they become colonized by these fungi to some extent, but we don't really know what they're doing, how much benefit they're providing. In the glasshouse, we've seen that these mycorrhizal fungi can provide a plant with all of its phosphorus uptake. So 100% of that phosphorus is going up through the fungus before it gets into the plant. And there's less data, but it's looking pretty convincing that they can be really important for nitrogen contribution as well. And they've got this sort of disproportionately high impact on the habitats in which you find these fungi. So they've been called ecosystem engineers. You know, they've got this really big impact. Given all this, I think there's like a a real opportunity for using them in sustainable agriculture. It seems kind of mad that we wouldn't try and use them, seeing as they're everywhere, they're engaging with the crops anyway. We might as well try and optimize that. But how can we do that so that we can reduce our inputs, make farming more sustainable? I think it needs three additive sort of components here. We need to make sure we've got the right genes in the right crops and that they're in the right soils. And that's where we come in here today with the right soils. But starting off with the right genes, a few decades of work have shown that there are a few key plant genes responsible for signaling between the plant and the fungus. And you don't need to pay too much attention to this diagram on the right-hand side. It just shows a sort of schematic of the sort of signaling pathways that are going on between the plant and the fungus out in the soil here. Especially important, we found, are these NSP2 genes. And by engineering plants so that they have different expression, different activity levels of these genes, we can control the amount of mycorrhizal colonization. So on the graph on the left here, you can hopefully see that what we've got is the amount of colonization in barley roots. And this is in a wild type plants, this is just commercially available barley seed. And under low phosphorus, you get pretty high mycorrhizal colonization. And under high phosphorus, where the plant kind of doesn't need to engage with this mycorrhiza because it's already got lots of phosphorus swimming around the roots. And so it kind of rejects the mycorrhiza in this high phosphorus case. And so you get lower colonization. But if we engineer these lines so that they're overexpressing that NSP2 activity is much higher, it alters the signaling between the partners and the fungi still colonizes. The roots are still receptive to that mycorrhizal colonization. And so both at low phosphorus and at high phosphorus, we've now got much higher mycorrhizal colonization in our roots. And the logic goes that we have more mycorrhizal colonization and we have more benefits. And we've tested it in the glass house, and we're now moving out into field trial testing, which is detailed in the article that Tom mentioned. And it's the first couple of years of doing this trial, and we've started very small. Because they're gene edited and genetically modified lines, there's a lot of sort of regulation and stuff we need to do for DEFRA to make sure we're all doing everything by the book. So the gene edited and genetically modified plots here are just these small chessboard sort of squares in the middle. Everything else is a wild type barrier just around the outside. And these are all happening in Cambridge, and we're testing lines that show enhanced mycorrhizal colonization, but also lines where we've knocked out the plant's ability to form these associations. And so if we're comparing those plants with the wild type, with the normal barley, we can see how much the mycorrhiza is doing naturally, and then also what it can do if we enhance it, if we boost that amount of colonization. And we're using NIAB's Park Farm site for this, and we found some soil with really low phosphorus, and then we're adding phosphorus into half of them so that we can test the effect there of high and low phosphorus. And as I say, small plots, it's not a yield trial, and we've just started, so I can't tell you much more about this, I'm afraid. So moving on from the right genes into the right crops. This is about mycorrhizal diversity, and this is sort of the wider genetics of more kind of nebulous genetic underpinnings here. So mycorrhizal colonization is definitely essential for mycorrhizal benefit, which makes sense, but it doesn't guarantee crop benefit. And this is because of a tricky problem where we've got a continuum from parasitism to mutualism, and not all plants receive the same benefit from mycorrhizal colonization as each other. And so I've got some data here from some wheat lines that I grew from the Avalon and cadenza population. So they're very closely related wheat lines here. And thus the height of the bar just shows their response to inoculation. So I grew them without mycorrhizas and with mycorrhizas and see how their growth compared. And hopefully you can see that these 4 bars on the left, lines A to D, there's actually a growth penalty when you grow them with mycorrhizas. The mycorrhizas are harming their growth. In some cases, really dramatically, a 30% reduction in shoot biomass. On the other side of the graph, We've got 4 lines which have a really strong beneficial response to the mycorrhizal. So there's something going on that means that these lines respond really badly to mycorrhizas. These lines respond really positively. And I don't know if you can see the scale, but in some cases, we're getting almost double the shoot biomass when we inoculate with mycorrhizal fungi compared to without. Annoyingly, we still don't really understand why this is happening. It's important to find out because, as I said earlier, all of our crops are mycorrhizal. So when we plant them, they become colonized. We need to make sure we're planting crops that are going to get a benefit from these fungi rather than be parasitized by them. And then lastly, the right soils. So it's looking increasingly like there's sort of increasing amounts of evidence to say that abundant and diverse mycorrhizal networks are the most beneficial for crop growth. We don't know that that is always true. We don't know how abundant or how diverse these mycorrhizas need to be to bring our benefit. So that's something that definitely needs work. Something that does seem pretty clear, though, is that the more disturbance you cause to your soil, the more you reduce that abundance and diversity. And that can be chemical disturbance with different fertilizers or pesticides, as well as physical disturbance, or call it biological disturbance. But I think this could mean, most relevant to this would be having bare soil is not good for mycorrhizal abundance diversity. It wants living roots throughout the year. So our soils have got mycorrhizas, but are they beneficial? We don't know. And are there safe levels of disturbance where these mycorrhizal benefits can be maintained? I think it's all very well saying that disturbance is bad for mycorrhizas, so don't disturb the soil, but you can't crop a soil without really disturbing it in some way. So trying to find a balance between disturbing it enough to get a crop, but not disturbing it so much that you're knackering the soil biology. And so proposal for a project, and this is all very preliminary ideas, so definitely want feedback from everybody on this. I'm proposing a sampling survey of UK farms, and I want to find out how much mycorrhizal fungus is in UK farm soils. How does farm management affect this? So different practices, whether that's no-till or cover cropping or regenerative practices or organic certification. We don't really know how this affects the mycorrhizas. And then also, are the mycorrhizal fungi the same according to their location in the country, their management, the management of the soil, but also soil type, crop type, and so on. And so I think to start in a kind of controlled way, we need to standardize things. And so my suggestion would be that we use the same crop everywhere. So all the samples are from winter wheat, for example. And quite important that the samples are taken at a similar time, and that should be before flowering, which is where we get the most colonization. That's where it peaks. And then the sampling method, as well as analysis methods, should be as similar as possible, so we can standardize these results. And that's all to be confirmed. And so part of the plan is that I would ask you as participating farmers to collect root samples and then return them to the crop science centre by post. So we'll send out a sampling kit to everybody who wants to participate, and in there you'll have tubes of storage solutions and things like this, so that you can keep the samples safe, and instructions on how to sample and prepare all these roots. And then once those roots have come back to the crop science centre, we can quantify the mycorrhizal fungi in your crops and tell you what's there. And I don't want to overpromise or put too much on myself, but if you want to test different fields, different crops, we could probably do that as well. And then it might be a bit tight for time, but my plan will be to present the first of this data at the groundswell meeting in June. And I don't have funding for this yet, but what I would also like to do is to do some DNA sequencing on these roots, and then we can start to look at which fungi are there, how complex are these networks, and how does the soil management and location and all of this, how does that affect which fungi are there? And then we can start looking at sort of what are they doing? How are they influencing our crops? Are they a benefit? Are they not? And why? So that's my pitch. Thank you very much for listening, and happy to take any questions.
Great. Thank you very much for that, Tom. But before you go, and before I introduce the two farmers, we have had a couple of questions in. I just wanted to raise one of them, which is to do with oilseed rape. Thanks to David Fuller-Shapcott and also Tom Pearson on this. So we know that oilseed rape doesn't associate with AMF.
That's right.
Just give us a little bit about that, and also, whether by bringing in companion crops, as a lot of people are doing, whether that can sort of compensate, if you like.
Yes. So, as a brassica, oilseed rape doesn't form mycorrhizal associations, and it's still not really understood why. I think what's happened is that going back in evolution, basically all plants were mycorrhizal. And some families have lost that ability to form that association. And the current best thinking is that they found other strategies for getting phosphorus from the soil, whether that's secreting enzymes or acids that improve the phosphorus solubility in the soil. And so they can bring phosphorus to them in that manner. But it's still not totally understood why they don't form them. In terms of companion cropping, yes, I think definitely there is the potential for mycorrhizas to sort of indirectly boost brassica nutrition because they're being supported by these the companion crops or intercropping. And I have seen data that's showed that oilseed rape does directly benefit from nitrogen uptake from mycorrhizal fungi, even though they can't colonize. So the mycorrhizal hyphae sort of wrap themselves around the root and they just release some nitrogen and some phosphorus, and then the plants pick that up. So you're getting indirect benefits of having lots of mycorrhizas in your soil, even in plants that can't form that association.
Great, no, thanks for that, Tom. Now we've got, we've got loads of questions. Thank you so much. Keep those questions coming in. We're not going to look at them now. One thing I was going to say is just going back to what Tom was saying about how we're going to run this trial. So I'm going to be working closely with Tom to develop a protocol, a standard protocol that we can bring to those people who are going to be involved. If you remember when you registered for the webinar, you all gave some information about your cropping systems, and thank you so much to those of you who filled it in. I can tell you we've got a really nice spread across the country, across soil types, across systems of people who'd like to be involved. We won't be able to involve all of you, I'm afraid. That's very much limited by funding. But what we'll do is we'll get in touch with all of you, check that you still want to be involved, and so on. Really, the number of samples we do will come down to funding. And that's another point Tom mentioned about funding. We have been looking, since we first came up with this idea, we have been looking for funding. And the more funding we can get, the more sampling we can do, the more we can make of this project, and the more we can really make it mean something. So I know that there are a lot of you who have got funding pots available watching this webinar. And if you haven't personally got a funding pot available, then nudge someone who has. And get them to be involved. Get in touch with me, tom@boffin.org.uk, and obviously we'd be delighted to have some funding on board for this. Anyway, I'm going to crack on now, and I'm going to invite Simon and Tim to turn on their cameras and to join the meeting. So Simon Cowell farms in Essex, and Tim farms in Staffordshire. Thank you very much for joining us, guys. And as you probably aware, they're also founder members of the Green Farm Collective as well, and have been pioneering regenerative practices on their farm. So Simon, first of all, could I just ask you to give a little brief introduction about your farm and your system?
Yeah, thank you, Tom. I farm on the east coast of Essex on low-lying sort of marshland, mostly heavy clay. 20 years ago, I was struggling with seed beds and things, and I went over to direct drilling, and it's just been the best thing I've ever done. And the soil conditions just improved dramatically since I changed, so that now I've got beautiful soil structure, just looks like chocolate cake. And that's due to not disturbing the soil, but also it's built up that way because of the biology and the mycorrhizae that have created that soil structure.
Yeah, no, tell me about that, Simon, because I know you've done quite a few explorations into that. So tell us a bit more about your sort of journey with AMF, if you like.
Well, once you get onto this regen system, you start being fascinated by soil biology and learning about it and understanding what it can do for us. And quite a few years ago, I started sending off roots to be analysed, and it was really quite interesting what we found out. If you put my slide up for a start.
Sure. Hang on a second, bear with me. I will do that now.
We did. I always have a variety trial, a lot of different varieties in strips, just to see how they perform on a sort of my system. And we did this analysis of roots. So you can see the blue lines were roots collected in March, and then in July, orange lines for each variety. And when I started, we used to think that sort of 30% colonisation for cereals was pretty good level, but you can see we're up to 80% on some of these. And this is as a result of the way I'm farming, of no-till and not applying any phosphorus fertiliser, sort of forcing the plants to associate and find their own phosphorus. But my only worry is whether this is too much mycorrhizae now, and these are actually becoming sort of like parasites on the crop, because I don't really see any increase in yields from having more mycorrhizae.
Well, no, it was interesting, the that data that Tom showed earlier, and perhaps that's something that we can explore either within this project or perhaps another project. Tell me more about what you've been exploring, Simon.
Yeah, I mean, like you said, I thought that was very interesting, Tom's slide about the biomass response. We need a lot more to learn, and maybe I've got the wrong varieties that we could find out which were the best ones, might make a difference. And if you just put my other slide up, this is I grow certain amount of lucerne on the farm, which is a brilliant crop for general soil biology. It's in for 3 years, but this was quite interesting because we took a root from a lucerne plant that was only 3 weeks old, and straight away you can see in the slide that completely covered in the blue stained mycorrhizae just 3 weeks after it's come up. But the interesting thing is that the field was just a bare overwinter stubble for 7 months, no living roots at all, and yet this is colonised straight away. And I think that with my system of loads of mycorrhizae there's a huge amount of mycorrhizal spores in the soil, and that they were able to sort of attach to the roots straight away. So I'm not sure that you really do need living roots all year round once you've got the system working.
Yeah, no, absolutely. No, thank you very much for that, Simon. I'm going to ask Tim that do you mind if I stop sharing the screen now, Simon?
Yeah, fine. Yeah.
Tim, can I ask you to come in now? Just tell us first of all a little bit about your system.
Yeah, morning everybody. So I farm in South Staffordshire. Obviously been doing regen since 2009. I'm 50-50 winter to spring cropping, growing milling wheat, winter oilseed rape, spring barley, spring beans, spring lupins, corn marigolds, corn flowers, chamomile. I'll have a go at anything. I don't use insecticides, haven't done for 8 years. I don't use fungicides, don't use growth regulators. I don't use muscicides very often.
Yeah, no, that's really interesting, Tim. And I hadn't actually been to your farm. It's one of my my aims is to visit your farm shortly. But I have seen and I've read a lot of what you said about this, and that you don't need to put these inputs on. So how are you managing to do that? I mean, what's been your journey? What's your secret here, Tim?
With insecticides, with aphids, it's the sugars. So the plant isn't synthesizing the sugar. So by putting the correct nutrients down there, the plant, because I put a lot of nutrition down at drilling, especially with oilseed rape. So the plant is producing monosaccharides and monofructose. By having the right nutrition there, the plant can then convert those sugars into polysaccharides and polyfructose, which will then go down as exudates. Once the plant gets them into a polysaccharide, the flea beetle, for instance, can't digest them. So it's the wrong food source. When the flea beetle are attacking the plant, it's because the plant isn't synthesizing properly, which is why the flea beetle are coming in. It's just nature's bin meant to take an unhealthy plant out. So that's how I get around that one, and the same with wheat and barley with BYDV. It's just having the plant working properly.
Right. So, and what role do you think the soil biology then is playing in this, and specifically the fungi there then? Because the soil isn't as oxidized, because I haven't moved it, everything's more available, so that the plant can take in those nutrients far easier, which is why I'm producing a healthier plant. I'm not using anywhere near the nitrogen. So last year, my best crop of wheat averaged 12 tonnes to the hectare. That was with 50 kilos of soil applied in and a little bit of foliar, 28 litres of foliar nitrogen. And by doing that, I'm not damaging my rhizosphere, my soil, because I'm not putting all those inputs on which are burning roots and killing biology. I'm trying to work in the heartbeat of nature. And by doing that now, everything is becoming available, and that's why I'm getting the yields. It's just, once you get the system working, the benefits are fantastic, but you've got to earn the right to get from A to Z. You can't just do it. I have too many people saying I've stopped using fungicides, but I had a 2 tonne to the hectare loss. Well, I do lots. I don't just stop. I'm doing an awful lot of work. My management is probably 5 times harder than a conventional system. Yeah, no, that's interesting that you say you've got to earn the right, and you're not the only person to have said that. I've heard that a number of times. But when it comes to this project itself, Tim, and thank you to you and Simon and everyone else in the Green Farm Collective for wanting to be involved in this, by the way. But what are you hoping that this project is going to show us? What are we going to learn? Well, mycorrhizal fungi is a fascinating subject. Most of my soils are either 1-to-1 to 2-to-1 fungi to bacterial. Once you've got that mass of mycorrhizal fungi, you could have 10,000 times the root mass to help feed that plant, to give it water. The largest mycorrhizal fungi, as I'm aware of, is 2,000 square miles. It's 2,500 years old. This has been working for millions of years. It's us as human beings that's got in the way and caused the problems that have stopped this system working. It was already working fine before we came along and said, right, we'll take it from now. We know what we're doing, and we didn't know what we were doing. It's nobody's fault. We've just created an awful mess, but it's now time to rectify that situation. Right. And how about yourself, Simon, if I can ask you to come back in? What are you hoping to find out from this project?
I think pretty inconclusive what I've found out, and I just want some more research done in a more scientific way, really, to try and find out when it's an advantage and when it's a disadvantage to have more mycorrhizae. And I've got all sorts of different situations on the farm where I've got fields that I haven't put any nitrogen on, to see what difference that's made and whether they're working differently or different strains have evolved, and masses of things really we need to find out. Yeah, no, absolutely. Well, I'm going to ask Tom if you can come back in, and we've got loads of questions coming through on the Q&A. Thank you very much for those. I'm going to try and actually get people to join. Well, one question we've got is from Keith Norman. Keith, would you be happy to actually join us? If you'd like to join us, if you put your hand up, I think that'll work. I'm not entirely sure how this works, if I'm honest, but we'll give it a go. And I think I'm not entirely sure this is going to work, by the way. I can't find you, Keith. Wait a minute. There you are. Oh,
Blast, hang on, allowed to talk. Are you there, Keith? I think you need to you need to unmute yourself.
Yeah, I'm unmuted now. Is that working?
It is working. Yeah, Keith, you've got a question.
Yeah, it's really just obviously we're talking a lot about natural fungi in soil, and a lot of things that we apply to the soil could be detrimental. And I'm just wondering if any studies have taken place looking at the impact of things like residual herbicides and nematicides on those, mycorrhizal populations, and what's the knowledge in that area?
There's a study out recently from quite a big project in Europe that went through, I think they sampled from the Netherlands down through Central Europe and into Spain. And I think the general trend that they saw was that the more you add, the worse it is for your soil biology. The higher number of chemicals, the different classes of chemicals, and the rates at which you apply them, all of that just adds up to, it's an extra stress on your soil biology. And so there will definitely be some that are worse than others. And I think there's a lot of work going on to work out which are the best, which are the most specific for their target. But generally the message seems to be that more chemicals you put on, the worse it is for the mycorrhizas.
Yeah, that's no great surprise, really. And is there any evidence that the levels actually deplete to a base level that they can't recover, or are they always quite resilient? And when the pesticides stop, they recover and their populations start building again?
Yeah, they definitely do recover, and they recover in a number of different ways and different sort of timescales. So if you till the top 15, 20 centimeters of soil, you're going to get recolonization, I think, mostly from below. So they will sporulate in that top layer of soil, but you've still got a hyphal network in your lower soil profile. So the fungi will come back up. In terms of the communities, when you're sort of knocking them back, yes, they can recover, and their best chance of recovery there is if they are close by to unmanaged grassland or woodland, where you've got that bank of mycorrhizal fungal species that can infiltrate and grow back in.
Great. No, thank you very much for that, Keith, and thank you, Tom, for your replies. Now, Tim Ashley, I noticed you've got a question in the chat there. If you'd like to raise it yourself, do you want to stick your hand up? There you go. And let's see if this works. Hang on. Can you join us, Tim?
Yes, I'm here. Can you hear me?
Brilliant. Yeah. What's your question, Tim?
Well, it's really interesting to see the spread between the parasitic and the symbiotic mycorrhizal, and I was wondering whether, as part of the surveys or the analysis, that you look at the wider communities that are in the soil, like Tim Parton mentioned, having his soils a fungal bacteria ratio of 1:1. If we've got decent diversity of the bacteria, I'm led to believe that in order for the AMFs to kind of colonize, they need the signals from other biology in order to grow and colonize. So I'm just wondering, do we need to sort of look at the wider soil food web in terms of these studies? That's it.
Yeah, I'd say so. I think you're absolutely right that they're all going to be playing a part, and we can't just look at the mycorrhizas and think we've solved it by quantifying them or identifying them. And absolutely, it's the sort of thing that you can do. If we take samples for DNA sequencing for the mycorrhizas, you've also got your bacterial DNA and your everything in there, tiny arthropods, all sorts of things that are engineering our soils and crop responses. So yeah.
Great. Does that answer your question, Tim?
Yeah, I think so. I'm just really interested in the whole soil food web, and how everything interacts. I'm blown away by the fact that viruses are inside bacteria, bacteria are inside fungi, fungi are inside the roots. It's all of these things are interacting, and sort of measuring them and seeing, you know, what the diversity is in these soils can maybe lead us to understand, you know, how we can best use the mycorrhizal fungi to our benefit.
No, indeed, absolutely. Now, moving on, Tom Pearson, if you'd like to ask your question, do you want to raise a hand? Are you there, Tom? Fantastic. Hang on, let me just bring you in, and then are you there, Tom?
Yes, I am. Sorry, which question did you want me to ask? I asked a few as per usual.
There was a question here. For Simon about home saving seed, which I thought was an interesting one.
Yeah. Sorry. Hi, Simon. Yeah. So just I suppose I was, you know, you had that graph looking at your cereals that you said you would thought 30% would have been good, and you've now got up to 80. Are you managing that through home save seed and the fact that you're getting this sort of terroir effect on your cereals? And then the other sort of part of that question maybe might link in is, I know you're a bit of a compost king, so I'm wondering what your sort of compost work has, how that sort of improve your soils, and whether you think that's a key part to you managing these high AMF associations with not so many living roots as other people might use.
I mean, composting is just a small part of it, I think, really. Obviously, putting on compost is fantastic for the soil biology. It's sort of a really good inoculation. But if you did everything else and don't disturb the soil, the mycorrhizae would build up, I'm sure, just as well. That's just a little extra help. Yes, I've mostly used home saved seed without seed dressings. Again, it's not essential. It's just another little thing to add that helps, I would have thought.
Yeah, has that answered your question, Tom?
Yeah, absolutely. I just think it's really interesting to consider home saved seeds. Obviously, you've got your own population of mycorrhiza within your farm, and then you bring in bought seed that's got different, maybe perhaps used to, it's a bit of a layman's terms here, but used to different associations, and therefore you're sort of you're not, your home saved seed really can get going quickly because it's used to the associations that are already in your farm, perhaps.
It's another thing. It's the sort of thing we presume. We don't really know that, and that's something else we'll need to look into and find out, really.
Great. Now, there's a really interesting question that's come through from Anne Osborne. And if you want to raise it, do you want to raise your hand? If you're there, fantastic. If I can ask you to come in.
Morning.
Brilliant. Yes. Now I was talking to Anne yesterday, actually. So and you're doing some really fascinating research in this area. So do you want to raise your question, Anne?
Yeah. So you were talking about artificial chemicals and pesticides, but one of the things that we work on are the molecules that plants make naturally. Oats, for example, make a chemical in the roots called adenosine, which is antifungal, and we've shown that it protects oat roots against infection by the take-all fungus. I've also had discussions with Keith Norman quite a while ago. We even wrote a grant proposal about the possibility of oats controlling blackgrass. So it's interesting to think what these natural chemicals might be doing in the soil and in crop rotations, both for AMF and other soil microbes. Very, very interested in anybody who would like to get involved in that at some point. I think this is a fantastic group.
Tom, that's a really interesting point from Anne. Could I ask you to, I don't know whether you've done any work on this, but of course, I mean, you talked about the pathogenic side of AMF, but this other aspect that Anne raises, that, you know, perhaps the systems that plants use to fight off fungi like take-all could actually fight off AMF. Do we have any evidence or anything like that happens?
Yeah, it's a really interesting point. You know, if we are adapting our crops so that they're much more receptive to a certain type of fungi that's beneficial, we don't want to be letting in those pathogens. We're just opening the doors to anything to colonize the roots. I haven't done any specific pathogen trials yet, but it's looking like our work sort of making plants susceptible to mycorrhizas doesn't make them massively susceptible to disease, which is a massive relief. I should say as well, in this trial, just because of the variety we're using for barley, it's susceptible to mildew. So we did have to use a fungicide on that, a preventative fungicide, and it doesn't seem to have had much effect at all on the mycorrhizal colonization. So that's encouraging as well, that, you know, it's not, if you use fungicide, you've got no mycorrhizas. But in terms of, plant secondary metabolites and things like this, chemicals they're producing, I think there needs to be a lot more research done on that to see how it's affecting all of these positive and negative interactions.
Well, that's interesting you say that, and I might have something to say about that at the end of this. Moving on, David Fuller-Shapcott. David, you put quite a few questions in to the chat. Would you like to come in? If you want to raise your hand. Hang on a second. Let's just bring you in. One of the things that you're talking about was seed dressings. I know we talked a little bit about home saved seed, but you've got a question on seed dressings in particular.
Morning, Tom. Can you hear me all right?
Yeah, absolutely.
Excellent. Right, thank you. Yeah, I'm struggling to get my head around the idea that if you put a fungicidal seed dressing on a seed, stick it in the soil, that it has no effect on the fungal populations. Now, I asked this question of an eminent professor of plant pathology that you and I both know up in Edinburgh, and she concluded that there probably wasn't an effect, but I'm wondering whether the panel would like to just comment on that.
Yeah, Tom, do you want to kick off on that one?
Yeah, well, I'd be surprised if there was no effect at all. I think early colonization by mycorrhizas is bound to be hampered if you've got a fungicidal seed dressing. I guess the hope would be that the roots are going to grow out away from that zone where that fungicidal seed dressing is, into soil that's got mycorrhizal propagules in it, and so colonization can take place there. I know that seeds that have had a dressing, fungicidal dressing, those plants do go on to become colonized by mycorrhizas, but in terms of what effect that dressing has overall, I'm not sure. I don't know that much work has been done on that yet.
I don't know whether it's Simon or Tim. Have you got any thoughts?
I mean, the variety trials I did, some of the seed was sent in with dressing and some without, and I couldn't say that there was any difference really in the end result. There was nothing obvious to say differently. No.
So I think where I used to buy in seed, because I'd ask seed to, but if I was buying a new variety, I'd ask seed to be undressed, and inevitably 5 years ago it would still come dressed. You can buy it now undressed, but where I planted the same variety, the same plants per square metre, the germination difference was tremendous. I've got a good slide when I put in my talks, and you can see that the plant is 10 days in front of the dressed seed. It germinates quicker, it establishes the root mass was bigger. It had a massive effect here when I had a fungal dressing. And the other thing where I've stopped using fungicides, if I chop the straw, the breakdown of that straw is 10 times faster than if it's had a fungicide on there, because you put that barrier to stop fungi breaking that straw down, in my opinion.
Well, that's great. Listen, thank you very much, everyone. I'm going to draw the discussion to a close now, but we've had some really, really interesting thoughts and ideas come up, and comments as well. This is really, really good. I've got your questions in the Q&A, and we'll go through those, and I'll try and get answers to those ones that we haven't addressed. But what I would like to do is that there's a funding round that's up at the moment, and I just wanted to say a few words about that and whether there's an opportunity here for us. So Thank you very much, by the way, to Tim and Simon and to Tom, of course, for your contributions and for coming here today, and especially to Tom for that excellent presentation. And as I say, we'll come back. I'll come back to all of you with these questions as well. No, I just want you. I'm just going to share my screen quickly. So a lot has been said about soils, and many of us are drawn to books such as Dirt to Soil and to legends such as Gabe Brown for our inspiration. But where is the scientific understanding? Now, nothing against what these great authorities bring, but it's largely built on their own observation and practice. So how repeatable and measurable is it? And that's what I think we should be looking at. So what is the truth about, this is a quest to discover the truth about soil. I've got some questions, and here they are. They're sort of pretty fundamental. Hang on a second. Some pretty fundamental questions. What I'd like is your view on these questions. And so if I can get the technology to work, I'm going to get a poll going in a minute. But these are the three pretty fundamental questions about soils that I think we need more answers on. What's the optimum root architecture? What role do soil microbes play in feeding the crop? And how do you manage soil and root pathways? Now, we've got a poll. I'm just going to see if I can get the poll to come up. So bear with me while I try to get the thing to work. So hopefully you should have on your screen that question. So which of these are you most keen to know more about? So the optimal root architecture, the role of soil microbes, or management of the soil and root pathogens. Now I'm not entirely sure how we display the results here. Yeah, no, we've got some really interesting ones. I don't think it'll display the results until I end the poll, but I can tell you that the role of soil microbes is very much leading here. No, thank you very much, everybody. I'm going to end the poll now, and hopefully it'll display the results. Yeah, there we go. As you can see, most people going for the soil microbes. We've got 22% looking at soil root pathogens and 9% the optimum root architecture. So thank you very much for that. Sorry, bear with me. Trying to run about 5 different things at once here. So the other element of roots is the interventions, and we talk quite a bit about that, whether they're positive interventions or negative interventions. Here are some interventions that I think, we apply as farmers. One of the main ones that really we've got a lot of questions about is just what's actually going on under there. How can we measure it? How can we quantify the amount of root mass we've got, the architecture we've got, and assess that, and how beneficial, what's it actually doing to our crops and to our soils. Measuring and benchmarking that. Stimulation, and the inputs that we apply, and it's particularly sort of biostimulants, but also fertilizer and nutrients and so on. What are they doing? And also, bio inputs as well, not necessarily synthetic inputs. Genetics, the role of genetics here, and we've heard a lot about that from Tom, and there are traits coming through. Just so what traits are actually useful, what traits can we use, and how can we use them? Cultivations, there's quite a lot of work that's gone on here, but just how much do we know about how the cultivations in different scenarios and situations affect the roots? And then there's companion cropping as well that I put in here. By the way, in cultivations, I include cover crops, for the regen ag guys and for those people who aren't, doing any cultivations. Obviously, there's roots as well as iron that you can do cultivations with. But companion cropping is different. We're talking here about diversity, and how co-cropping and companion cropping can benefit the actual crop roots. So again, I'm going to try and get a poll going. So bear with me while I try and get the next poll. Right, okay. How do I do this? Download results. No, don't want to do that. Sorry. Yeah, this is interesting. I can't I can't seem to get the end poll. Can't seem to get the next question up, unfortunately. Listen, sorry about this. We're gonna have to move on, unfortunately. What I'm hoping, sorry, what I'd like to do is get a project going, and what we could draw up is like a matrix of truth, if you like. So you've got the questions on the left-hand side there, and then the interventions along the top. And this really to illustrate what's going on, and to show that, you know, that there is some good science going on and projects that are here. So you can see these sort of spheres of activity. And this is just illustrative, by the way, as to how much is actually going on. So not necessarily definitive projects there, but just to give an idea. What my point is that, you know, how much of this is actually filtering down to on-farm practice in a measured and measurable way. So this project would aim to do that in a way that has clear on-farm deliverables. So first of all, a series of work packages that identifies current projects and plugs the gaps in our understanding. And then the on-farm trials, which would be a really, really important aspect of this, to test these interactions and to look at these interactions that we were talking about earlier on. And so what I hope that we'd end up with is firstly some really reliable and practical tools that we can use to measure root architecture, soil fungi, soil carbon even, you know, accurately and repeatably. And then we'll also have a much clearer understanding of what these products, of the products that we use, you know, whether they're synthetic or biological. We've had quite a bit of discussion about that today. And what are they actually doing? So these aspects of interactions as well, a really important one. And so what's the relationship between take-all and beneficial soil fungi, as Anne was saying earlier on, for example. And finally, a genetic toolkit to bring these findings into varieties that we use. So in future, we'll be looking at the AHDB recommended list for a wheat's rooting score or drought resistance, as much as we do for its septoria rating, for example. So my question to you is this a project that BOFIN should do and would be up for helping? Would you be up for helping with on-farm trials? And again, I was going to bring a poll up at this point, but I don't think it's going to work, unfortunately. But I really, really do want your feedback on this. You know, if you think I'm going down a rabbit hole on this one, then please let me know. If you support it, then please get in touch. And I've got my contact details at the end, but tom@boffin.org.uk. Do get in touch and let me know. And I'd be welcome any discussion around this topic as well. So look, I'm going to wrap this up in a minute. Oh, hang on a second. I do just have one final slide. Yeah. So the do go on to Trinity GFP, gfp.global, we're now, I think the page has just launched for this project, so it's on there, so please do join and have a look. It's a really great place to bring a lot of these thoughts and discussions together around BOFIN projects, so do get involved in that. Look out for all the news that we've got. As I said at the beginning, we've got some really exciting stuff going on, and obviously, you know, and we'll come back to you on this project as well. Thank you very much for your contribution so far to it. And that just remains for me to say thank you very much for attending this webinar. Our next webinar is on March the 16th. 8:30. The registration details are already up there. I will kick out another email about that. so that you can all sign up to that one. Really, really exciting on slug-resistant wheat. We should have some findings from the trials by then that we will be publishing for the first time. So thank you very much for attending. Thank you again to Simon and to Tim, and most of all to Tom for their contributions earlier on in this webinar. And thank you to all of you for your contributions and your discussion as well. And as I say, please come back to me and let me know your thoughts on this idea of a larger project that we could do. So that just remains for me to say thank you very much and have a great day, whatever you're up to for the rest of the day. Goodbye.